Okay, so we're going to talk about, let me enlarge this. Talk about mutations, changes in the genetic code. There are several kinds of mutations we're going to talk about. Mutations can be positive or negative. We'll talk about that. The major groups of mutations: frame shift, nonsense, silent, and missense mutations. Any change to the nucleotide sequence in the genome is a genetic code and is a potential for a driving force of evolution. And microorganisms, let me just move this. In microorganisms, mutations become evident in altered gene expressions, such as altered pigment production or the development of resistance to a drug. A wild-type organism is what its natural non-mutated characteristic is. The trait present in the highest number in a population would be considered the wild type of an organism. A mutant strain is an organism that bears a mutation, usually starts out as being few in number in the population, and then it can become a dominant trait. Let me just mark this person. So mutations, hold on, I have a couple of another person to admit. So mutations can be spontaneous or induced. For it to be induced, it's something that occurs from the outside that causes a change in the DNA. Whereas spontaneous mutations occur as errors that occur during, I'm sorry, more people are showing up. I will, when we take a break, I'll have to call the names of people that came in late. So spontaneous mutations are just that, the random changes that occur during replication. The induced are usually mutagens in the environment, chemicals, radiation, so on can cause mutations. Selected mutagenic agents, nitrous acid can remove an amino group from some bases. Ethidium bromide inserts between the pairs of bases. It's a dye, acrine dyes can cause a frame shift due to insertion between base pairs. Nitrogenous base analogs, they compete with natural bases for sites on the replicating DNA. All right, so some categories of mutation. Point mutation, that usually only affects a single base on a gene, such as an adenine, thymine, guanidine, cytosine is mutated. It involves the addition or deletion or the substitution of single bases. A lethal mutation is one that leads to cell dysfunction or death. Not all mutations are lethal. A neutral mutation produces neither adverse or helpful changes. A missense mutation is any change in the code that leads to placement of a different amino acid. A nonsense mutation is a change in a normal codon to a stop codon. that does not code. Remember, the stop codons do not code for an amino acid. So in other words, a protein could be cut short if a stop codon is, occurs somewhere in the middle, and usually that will lead to a dysfunctional protein. So categories of point mutations, and this shows you a frame shift or premature stop. A frame shift is where the code is read from a different position. Obviously, a stop codon will, as I said, lead to a shortened protein. A missense mutation in which the actual amino acid changes. The protein may not work as well. A base substitution, though, can also result in no particular change. If you remember, there's also redundancy in the genetic code. So sometimes a slight change in that third position of the code, you'll still get the same amino acid. A silent mutation alters a base but does not change the amino acid and has no effect. So that would be an example of that. The redundancy of the code assures that certain amino acids will not be altered by change in the third base of the codon.
Professor Fraser, can you go back to that last slide?
This one?
On the like the point mutations, there were some questions about the stopping and the starting that were based on that slide that I didn't quite understand.
Okay. Well, so let me start maybe up here. So this is the normal gene. So you can see the DNA codons up here. And you're right, it's not you, I shouldn't have skipped over that so quickly. So up here's so this would be the normal gene. These would be, remember, the code is found on the DNA. The codons are on the message RNA. There's AUG, which is the start codon. And this doesn't show a stop codon, but these are all the codons and what the amino acids are. So taking this one here, a missense mutation leading to a different amino acid. So a change in the DNA from G to T results in a codon that goes from GAC to GAA. And you can see in this instance, we get a different amino acid. And that's what we call missense mutation. It's the wrong amino acid.
Mm-hmm.
A base substitution, no change in function. So this was the original codon. Down here it becomes CTA. So in other words, the G changes. The codon becomes GAU, and you get a different amino acid. But if there's no change in function, I'm sorry, you get the same amino acid. Up here, you got a different amino acid. So even though the codon is different, remember there's redundancy in the code. What's important is the first two codons. The third one, we have what we call wobble. It can be different and still give us the same amino acid. So this is a silent mutation. It doesn't really cause any change in function. Now, frame shift, they're showing up here, is if you get a premature stop. So here, what we had is if you started reading at a different position and you got down to here, where you're supposed to have AUG get methionine here again, but here it gets changed because of the shift in the reading frame, it gets changed to a stop codon, UGA, that does not code for an amino acid. So whatever this protein is from here on down does not get translated. Whatever codons from here, and if there's other codons after it, they do not code for an amino acid. And this is just shows you another frame shift mutation here. So that can be pretty bad. You can either get a dysfunctional protein that doesn't work, or it's or other things leads to a premature stop.
Yep.
Okay, Jeremiah Kelly. I'll have to go over that later. A back mutation occurs when a gene that has undergone a mutation reverses to its original base composition. So occasionally a gene can mutate and then mutate back to its original base. Frame shift mutation occurs when one or more bases are inserted or deleted from a newly synthesized DNA. So this is occurring during replication of the DNA. So that change results in an altered reading frame for the message RNA. This almost always results in a non-functional protein. Sometimes when you have mutations, your body will try to work with the protein. If I remember correctly, collagen's made up of 3 proteins. 2 of them are the same, 1 is different. And the one that's different, there's some people will have a mutation, and what the body will try to do by compensating is they'll just use all 3. They'll use 3 of the same protein instead of they're supposed to have a combination of 2 that are the same and 1 different. And that results in a disease called osteogenesis imperfecta, where the people have weak bones. So anyway, that change in the reading frame almost always results in a non-functional protein. Every amino acid after the mutation is different from what is coded for in the original DNA. Insertion of bases and multiples of 3 does not disturb the reading frame. It's when you have bases, 1 or more bases, but 3 bases could give you a different amino acid at that place, but the reading frame would continue till it gets to the stop codon. Repair mutations. So you do have enzymes that work to repair mutations. You have repair of damage caused by ultraviolet radiation, and you have what's called excision repair. Mutations are removed by a series of enzymes. The incorrect bases removes the incorrect bases and adds the correct ones. So there's an enzyme complex 2, and here it removes these and inserts the correct ones. And this other example, they cut out the incorrect bases, and then the new bases are added in. So it's 2 steps. First, you remove the incorrect ones and then add the correct bases. A mismatch pair, a repair system can locate mismatched bases. So remember, bases have to, G always has to bind to C, A always has to bind to T. And so if the system sees that there's a mismatch, The base must replace soon after the mismatch is made, or it will not be recognized by the repair enzyme. So this has to occur very quickly. There's a test, common test used to detect if chemicals have a carcinogenic potential. It is called the Ames test. The Ames test uses bacteria rather than experimental animals. It allows for easy observation and monitoring of gene expression and mutation rate. Any chemical capable of mutating bacterial DNA could be similarly mutate mammalian DNA and is therefore potentially hazardous. So you take a culture of bacteria, for example, and you have them, let's say, not being able to make histidine. In the control setup, bacteria plated on histidine-free media containing liver enzymes but lacking the test agent. So it's a control plate, minimal media lacking histamine. Colonies arrive from spontaneous back mutation. They mutate back, in other words, they gain the ability. So a few of them will be able to express it. The experimental plate is prepared the same way, except that it contains a test agent. So now you have a plate with a chemical in it. The test chemical has no histidine. Colonies in the presence of the chemical mutate. And there you see that you can then see how the degree of mutagenicity of the chemical agent being calculated. You compare the number of colonies growing on the control plate, which is this plate, just has a few colonies. And you compare that to the test plate that has the actual chemical. So many mutations are not repaired. Effects of mutations depend on the nature of the mutation and the strategies available to the organism. Mutations are permanent, they're heritable, they're passed on the offspring of organisms and new viruses, as we've seen already with the COVID virus. First of all, it's an RNA virus. RNA mutates very easily. And there's a whole bunch of new variant of the COVID virus. There are obviously millions of people getting infected, and each person's getting infected with millions of virus particles. So statistically, the chances you're just going to keep having mutations. A small number of mutations contribute to the success of an individual in the population. Variant strains with altered ways of expressing a trait can maybe more readily adapt, survive, and reproduce, which is exactly what's happening with these variants of the COVID. The original COVID virus, they figure talking to someone who has it, it might take you about 15 minutes to get infected. Now with these new variants that seem to replicate quicker, it's a shorter amount of time before you get infected, being exposed to someone. Okay, this is the last slide. So true or false, a spontaneous mutation arises from exposure to chemicals or physical agents. Is that true or is that false?
False.
So that's false, yeah. The spontaneous mutations occur during replication. They're just that, spontaneous. So an induced mutation is exposure to a chemical, physical agents, that would be like UV light or X-rays. A blank mutation results in a stop codon.
Nonsense.
Or was that a frame shift? I always have trouble remembering these different ones.
Should be said announce.
So frame shift and a premature stop. That's what I was thinking. Oops, sorry, went too far.
It's a nonsense, I thought.
Well, I went back to the slide and they're saying frame shift. And so the reading frame changes and then a series of bases become a stop codon. Whereas a missense would be a change, a switch in amino acid. You can have a base substitution, but no change in function. Remember, we have redundancy in the genetic code that protects us. And I didn't mean to go so far. Let's go back for a minute. A blank mutation nearly always results in a non-functional protein. So if we just go back again. To look at these. So lethal mutation leads to cell dysfunction or death, but the in a missense was any change of an amino acid. Nonsense is a normal codon, turns into a stop codon, which could be also lethal. You wind up with a dysfunctional protein. So the Ames test is a first-line chemical screening technique. Oh, sorry, I answered the question. To detect mutagenic chemicals. True or false? Mutations are always harmful to organisms. Do you think all mutations are harmful?
No, some help with survival.
Yes, so some mutations could be beneficial. In other words, they could be the material for evolution. Obviously, the mutations that the COVID virus is undergoing right now are beneficial for the COVID virus, but not for us. We're now in a race to get people vaccinated before these keep mutating. Our country seems to be in a little bit better shape with that. Europe is having a lot of trouble. On the other hand, you have countries like Brazil. The leader of Brazil doesn't believe in wearing masks, social distancing. He was even for a while talking down the vaccine. He just believed in herd immunity. He's starting to shift on that because it's becoming quite a disaster. All right, so that's it for this section. Let's take a look. I just had a quick review of our last section that's in the module. So there were some definitions. All the genetic material in an organism is called the genome. Is that true or false? All the genetic information. Is that referred to as the genome? Nobody wants to take a shot at that?
True.
That is true. the definition. The genetic information in bacteria is found on one single circular chromosome and in plasmids. Is that true or false? So first part of that is, do bacteria have one single chromosome? Is that true? Nobody? Everybody leave? Do I need to unmute people? Okay.
That's true. It's typically a single-stranded DNA molecule.
In plasmids are also extra chromosomal pieces of genetic material found in bacteria. So that would constitute the genome of the bacteria. In eukaryotes, the genetic information can be found on chromosomes in the nucleus. It can also be found in the mitochondria. In plants, it can also be found in the chloroplast. Is that true or false?
False.
That is true. So in us, for example, our genetic information is in the nucleus, but we also have some genetic information in our mitochondria. Plants in their chloroplasts also have genetic information. So that is true. Number 4, the genetic code is found on DNA. Codons are found on the message RNA, and the anticodon is found on the transfer RNA. Is that true?
True.
Okay. DNA itself is made up of a phosphate. I'm sorry, somebody have a question? DNA is made up of just 3 things: a phosphate, a deoxyribose sugar, and a nitrogenous base. Is that true?
False.
That is true. Sorry, let me do that. It is just a lot of whites. The DNA is a very simple molecule, just 3 things. There's 4 kinds of nitrogenous bases. You should know what those are, and you should know which ones bind to which ones. So when it comes to DNA replication, which one of the following is not involved? Question 6, not involved in DNA replication. There's a helicase enzyme. These are all real enzymes. A DNA polymerase, a ligase, and an RNA polymerase. Which one of those is not involved in DNA replication?
RNA polymerase.
Yes, the RNA polymerase. So the polymerase is involved in transcription of RNA. Does anybody know what the 3 different kinds of RNA are? Question 7 kind of tells you that. So which one is not found in transcription and or translation? So you have message RNA, transfer RNA, you have ribosomes, you have amino acids, and DNA polymerase. So which one of those is not involved in transcription or translation? It's the DNA polymerase, which is involved in the replication of DNA. Which step or steps are part of protein synthesis? Initiation, elongation, termination, or could it be all 3? Okay, it's all 3, and they are in the correct order. When you start, protein synthesis consists of the transcription of the RNA and then the translation of the message RNA that occurs on the ribosome. It starts with initiation, then elongation, and then eventually it terminates. Can more than one codon specify for the same amino acid?
True.
That's true. Lastly, the lac operon is an example of regulation of proteins at the genetic level. Is that true? You remember the lac operon? That is regulation. It is occurring at the genetic level. That is true. All right, so we're going to go now and talk about DNA technology. Let me get rid of that slide. Okay, so we're going to talk about genetic engineering and recombinant DNA. Anybody ever watch a TV show called Designated Survivor? Anyway, there was a whole, it's about a guy becoming president of the United States because of a disaster. And he encounters one problem after the other, but there was some episodes about somebody genetically engineering the smallpox vaccine. That was very interesting and a little alarming if you thought about it. Very alarming, actually.
They don't have to do that. All they have to do is keep letting anti-vaccinators not get vaccinated.
Well, yeah, that's a huge problem too. They think anywhere from up to possibly 30% of the people in this country don't want to get vaccinated. And a lot of that's due to disinformation about the vaccines and a lot of misconceptions. And that means we're going to have a pool of a country of 331 million. 30% is a lot of people. And so they're going to be a ready reservoir for future generations of COVID virus. But hopefully hopefully we get to herd immunity and it stops the spread of the virus. I think we always know that there's going to be some people, for whatever reason, are not going to get vaccinated. So we'll talk about some modern genetic technologies, some of the things that we talked about previously with how bacteria get genetic information for either transduction, transformation, or conjugation. A lot of these techniques are used in modern genetic technology, along with some other things that have been applied. So genetic engineering is an applied science. A lot of these things have had basic applications. So some examples: genetic manipulation of transferred genes between a farmer's two largest pigs so that it's only advantageous genes are selected for. You can utilize DNA to identify a suspect in a crime. Fixing underlying genetic mutations to treat disease is a hot area of research. Utilizing RNA regulatory molecules to permanently fix disease. We didn't talk a whole lot about these other RNAs, but there are some RNAs that are involved in regulation. Basic science and applied science. What's the difference? Well, Basic science is just the research you do all the time. You don't necessarily expect it to pay off in the terms of medicine. Applied science, though, is taking what's learned in basic science and using it to develop medical cures for disease and solving other problems. Describe 2 scenarios in which genetic information can be manipulated in order to accomplish goals to benefit human beings. Talk about the tools and techniques of genetic engineering. A series of enzymes called restriction endonucleases. These enzymes are made by bacteria. They cut out very specific areas of DNA. You can use something called gel electrophoresis to analyze DNA. And it means almost exactly what it sounds like. You can put DNA in a gel material and subject it to an electrical current, and the DNA will migrate through the current. The bigger pieces of DNA will be at the top of the gel, and the smaller pieces will migrate further toward the bottom. Something very important, the polymerase chain reaction. It actually includes another step called RT, reverse transcription. This is how one of the ways we test for the COVID vaccine. I'm sorry, not vaccine, for the COVID virus. Where I work, they do what's called a pooled RT-PCR test. They do nasal swabs and pull them all together and test for the presence of the virus. And if it's positive, then everybody in the pool has to get retested to figure out who has the COVID. Describe how recombinant DNA is created. So anytime you put foreign DNA into DNA, it is a recombinant. And we'll talk about gene cloning. So DNA is the raw material. Some of the things we learned from last week, so some of the intrinsic properties. There's that enzyme helicase that unwinds the DNA. So in a laboratory setting, you can also just heat up the DNA and it will separate, and cool it back down and it will come together. So when DNA is heated to just below boiling, complementary nucleotides will hydrogen bond and will regain their double-stranded form when they cool down. So it gives you an opportunity when it opens up to put new genes into the DNA and using the restriction endonucleases. So these cut at very selected positions, so you can commercially buy these enzymes, and then you'll know exactly where in the DNA it will cut. And so they cut crosswise at selected positions. They're capable of breaking the phosphodiester bonds. So those, remember, the backbone of the DNA is that phosphate connecting the sugars. And between the adjacent nucleotides on both strands. These enzymes naturally occur in bacteria. It protects the bacteria from archaea and from bacteriophages, or potentially other plasmids. They can cut them up. Each endonuclease recognizes a sequence of about 4 to 10 base pairs. So you have a palindrome. So palindrome, at the end of the sequence, you'll have an area of bases that read one way and then on the other side the other way, but it's the same bases. It's called a palindrome. So they recognize those areas and make a cut. Once they make a cut, you have some staggered symmetrical cuts 4 to 5 bases on each strand, each pair with complementary tails. And you can insert DNA fragments or plasmids. So this shows you an example. So these are some examples of 3 different endonuclease enzymes: EcoR1, HindE3, HA3. And so it will make on the one strand, make a cut here, and it recognizes that this area, G to A, on the other strand, on the other side, it will cut in the same place. So you get this staggered cut. This one here just cuts right through it. So when you get that cut, you get what are called sticky ends when the strand comes apart. And then you can take another piece of DNA and put it in there. So the purple represents the original strand, and the blue, the new segment piece that's been put in to the plasmid. So remember, plasmids, or this, well, this could be the DNA also. Plasmids are used a lot.
Now, what is this used for?
This is, these enzymes can be used to put in foreign pieces of DNA into the bacteria or an organism. Usually in the lab, a lot of times you're working with bacteria, but you can put them into eukaryotic cells also. And you can either put a piece of DNA or you can put a piece of DNA in a plasmid and put it into a bacteria, for example. So restriction enzymes can create pieces of DNA, and these restriction fragment length polymorphisms are differences in the cutting pattern of specific endonucleases. So you get patterns of different lengths. So that way you can compare DNA of 2 different organisms at a specific site. So in other words, if you cut the DNA of one organism up versus another, you're going to see different fragments. And those fragments can, in forensic science, you can actually compare, because everybody has a unique, will have a unique signature of the fragments that get cut by the endonucleases. Some other things that are used in DNA technology, if you remember the Okazaki fragments that occur during replication, joined together by an enzyme called a ligase. We use that in the laboratory. So once you insert the new DNA, it can then be joined together after it's been cut by an endonuclease. The main application is final splicing of gene into plasmid chromosomes. Another important enzyme is reverse transcriptase. So if you have RNA and you have the reverse transcriptase enzyme, you can make RNA into DNA. This is what the HIV viruses do. They are called retroviruses. They have an enzyme. And and we'll see next week, after an infection with an HIV virus, what it does is it uses the reverse transcriptase to make DNA, and then the DNA incorporates into your DNA. COVID, for example, is an RNA virus. We can take advantage of that, because one of the tests we do, as I was mentioning, is the RT, which stands for reverse transcriptase polymerase chain reaction. So if somebody has the COVID virus, which is mainly messenger RNA, their genetic material is RNA, you can add the reverse transcriptase enzyme, make DNA, and then amplify it with a technique called polymerase chain reaction to identify the presence of the virus. And so we call that, when we take RNA and make DNA. That's reverse transcription. The DNA that's created is called a complementary DNA because it's complementary to the RNA, so going in a different direction. So for example, if we start with the eukaryote, we would have their, they have their DNA, but they make the RNA to make proteins, remembering that these introns that we have in eukaryotic DNA, not found in bacteria, the introns are removed. So you get the message RNA. It's going to be a little bit shorter than the actual genetic sequence after you remove the introns. And what we can do is extract that RNA. And remember, you're going to have in any cell, they're going to have thousands of different message RNAs that represent thousands of different proteins. You take all that RNA, you just simply add the reverse transcriptase enzyme, and it'll make a cDNA copy of all of these. So this basically is the same as this up here, but it does not have the introns. So this complementary DNA actually represents this sequence, which is called an exon, this middle sequence, and this other sequence. And then you can, with your complementary strand that you made, you can make a double-stranded cDNA editing enzyme. And remember, your gene now in the laboratory situation does not have the intron.
Good. No. It's close.
So in gel electrophoresis, you get a very specific patterns for DNA fragments when you cut them up with restriction endonucleases. And the gel is a soft agar gel that you can load the sample of DNA in, and then you put it under electrical current, and the fragments, because they have a charge to it, the DNA has that phosphate, they'll migrate in the gel to the positive end of the gel. As I said before, the bigger fragments will be at the top of the gel, smaller fragments will move quicker and they will migrate toward the bottom. The position of the fragments can be determined by staining the DNA in the gel, gives you a distinct pattern. So, for example, if you had some DNA and you wanted to look at the pattern, you'd treat it with these enzymes. They would cut it into fragments. You put it in a gel. This would be the top of the gel. You cast the gel so it has these little wells in it, and you can actually just, with a pipette, just pipette them right into the well. You then put it under an electrical current and you'll see a pattern. So usually you have some predetermined markers of certain size that you can use to calculate the size of these unknown samples. You add a dye to the gel that will fluoresce under UV light, and then you get these, you can see this patterns for, this is your molecular, molecular weight markers. You buy these from a company, and so they're predetermined what their sizes are. And then based on that, you can calculate the sizes of these unknowns here. Two different nucleic acids can be hybridized by uniting their complementary regions. So single-stranded DNA can hybridize with another single-stranded DNA or RNA. RNA can even hybridize with other RNA. And so we can take advantage of that to create gene probes. So a short stretch of DNA that's of a known sequence will base pair with a stretch of DNA with a complementary sequence if one exists in the sample. So you have an unknown sample, you have a known piece of DNA, and you look to see if that basically probing you're seeing is that sequence in your sample. So you have a carrier reporter molecules that can then with fluorescent dyes, so you can see, you can actually visualize where the hybridization is. You can add enzymes to the probes, which are then detected when a non-pigmented substrates become color molecules by the action of an enzyme. So you actually have a stretch of known DNA, and you create these with an enzyme linked to them. And then if it hybridizes to your sample, you add a colored dye that the enzyme then basically activates, and you'll actually be able to see where it's hybridized to the DNA. Uses of gene probes: you can diagnose the cause of infection from a patient's specimen. You can identify a culture of an unknown bacteria or virus. Nucleic acid hybridization does not require electrophoresis like we saw earlier. You take your DNA and isolate it, denature it, place it on an absorbent filter combined with a microbe-specific probe. The blot is developed and observed for areas of hybridization. Whoops. So you have a plate, let's say, of unknown bacteria. You then have your filter. You put that on there, and the bacteria adhere to the filter. And then the filter has a detergent that lyses these bacteria so that it will release the DNA. And then you can start to probe for it. As a gene, you add probes. They're labeled with a luminescent dye. And the probes will hybridize if there's a complementary piece of DNA in one of these colonies of bacteria. It will hybridize with it, and then it will luminesce under a UV light, and you'll be able to see where the which colony had the complementary region of DNA. There's another technique called in situ hybridization, fluorescent in situ hybridization. In situ means within a living organism. And this technique is called FISH, is the abbreviation. So probes are applied to actually live intact cells. They are observed microscopically for the presence of a genetic marker. It's a very effective way to identify genes on chromosomes. It's also effective in identifying bacteria living in a natural habitat without having to culture them. It can be used to detect RNA in cells and in tissues. Now, after this slide, we'll take a short break. Before we go on the short break, though, I want to call the names of the people I didn't get at the beginning of the class. So let me just go over this slide. The relative size of nucleic acids are denoted by the number of base pairs or nucleotides. So we've mentioned that the endonucleases recognize these palindromic sequences of about 4 to 10 base pairs. The average E. coli has 1,300 base pairs or 1.3 kilobases. The entire genome of E. coli is 4,700,000 base pairs, or 4.7 megabases. The human mitochondria is 16 kilobases. That would be 16 times 1,000. The Epstein-Barr virus has 172 kilobases. Our entire human genome is 3.1 billion base pairs on 46 chromosomes. All right, so we come back, take a 5-minute break, talk about polymerase chain reaction. I want to first, let me go back. Why does not let me enlarge? All right. So I'm just going to call the names. So please, it is Alexandra Tondia here? No. Is Brittany here? Is Kelly Butler here? No, I don't think she's here. Let's see. Ilika Demiki, sorry if I'm mispronouncing that. Are you here? I think Takea Dockets, I think said they weren't going to be here. You're not here by any chance? I think you sent me an email. Not in the class. Is Ashley here? Ashley?
Present.
Okay, thank you. Is Jermia here? Jermia Kelly? Not here. Jeremy Litton? Is Jeremy Litton here? Brittany Mobley, are you here? Dejaya Witherspoon?
Here, Deja.
Deja, sorry. Deja Witherspoon. Okay, so if I didn't, wait a minute, I think there's one person. Is Lillian here?
Yeah, Lillian is here.
Okay, thank you. So if I didn't call you, it's because I checked you in earlier. If you want to double check with me at the end of the class, you can let me know if you're not certain, but I tried to call the people I did not check earlier. So let's just take a break for a couple minutes. We have, oh, we have about, I think, 80 slides to go. All right, so I'll be back in just about 2 minutes. Yeah, so I got 8:05. Come on.
All right, so who's in Deborah's class? I need some help.
Here.
Here, I'm in Deborah's.
Some help in that lab.
I'm ready to. I'm ready just to be finished.
We all are. I still
I'm
haven't taken.
sorry.
I haven't taken my midterm yet because my brain is just like.
Me, I haven't either. I supposed to do it tomorrow.
Yeah, me
Yeah,
too.
I'm
definitely gonna take it tomorrow.
Yeah.
Dr.
I'm
Chester,
just-
look, I have a complaint. This lab ain't working out.
No, I know, but I'm
late to every class.
I don't understand like why we like where I don't like what is going on. Like
right.
We honestly, Professor Frazier, we 1 minute we just class is supposed to start at 7 and then it don't start till 8. And then we don't have class at all.
And we don't find out till 30 minutes after the class is over that we don't have class at all.
Our midterm review was only 30 minutes. So
Only
it's
thing
like
I could suggest with that is to talk to Dr. Malachy.
Oh, okay.
You know, I don't, you know, I don't know what's going on in the labs. I know it's a bad situation because normally we would have students in a classroom. Even this is not a very good situation to lecture. All you're basically seeing is my head, and in a classroom, I'd be able to move around. I could have a board that I could draw on. And it's even that's not an ideal situation, and the lab is definitely not ideal because the lab is designed to be hands-on. So it's very frustrating. This will probably be the last semester in which we teach this way, hopefully, but I can imagine it's very frustrating.
it's frustrating because it's like it's fast paced and it's like he'll get on and like I said, we'll have class from at least 7:30, 8 o'clock to 9. And with all that time we go over a quiz and then it's like a 15-20 minute lecture and then it's like y'all gotta know all this stuff and I'm not telling y'all the answers and I'm not gonna go over this and it's like, well, what?
And then the camera part with the test. Oh God, I mean, really?
My biggest pet peeve is that my class starts at 7 and I'm not waiting around and playing around in Canvas for you to decide to show up with class, and you're not even sending an email to let me know that you're going to be late.
Yeah, well,
that's one thing. If you send an email, that's just totally disrespectful to our time.
That I think is an issue you really should bring up with the department chair. You know, we should be here when the class starts and be here till the end. But definitely once it starts, if we get things done, sometimes we can end a little early. But I don't know. We have a responsibility to be here the whole time.
And that is very true. I mean, I got kids. I got a 1-year-old that I'm chasing around constantly. Like you can't start class at like 8 o'clock.
And then end like 10 because I have to get up for work.
Oh, right. Sure.
Right.
My only suggestion would be to talk to the department chair and try to engage the instructor, him or herself, to discuss it, which I know is, you know, maybe not easy, but you're the student and it's the instructor. We're here for you, or you're not here for us. You're not here, you know, you understand what I mean? So we should try to do what we can, given that it's a very not ideal situation.
Can I
All
ask
right, so
one
sorry,
other-
we got to get back to this. If you want to talk among yourselves, I can leave the Zoom open for a little while at the end.
No. Can you just answer one question for me, Professor Frazier? And that is, you had talked a little bit about the extra credit structure, because I feel like I'm going to need it based on my lab grade.
Right.
How will that be calculated into the final score?
Okay, so the McGraw-Hill, which are the homeworks and quizzes, that's 15% of your grade. The lecture that I'm doing here is 50% of your grade. So you have 5 exams. Each exam is worth 100 points. So if you look at it point-wise, each test is 100 points. We have 5 tests, that's 500 points. The McGraw-Hill, those quizzes and homeworks, 15%, that's another 150 points. That brings us to 650 points, or 65% of your grade. So the lecture's worth 65%. Is that clear? So then your lab is 35%, or 350 points. So in other words, everything adds up to 1,000.
Where does that extra credit come in?
Well, the extra credit is the number of assignments. There are more than enough assignments to get to 150. And if you did all the assignments, and I have had students in the past do every quiz that's there and every homework, I think it adds up to somewhere between 160 and 170.
Okay.
If you did every assigned quiz on the McGraw-Hill and did every assigned homework, it'll add up to more than 150 points.
Gotcha.
Now, do we have to do all the homeworks? Like, if we don't finish all of them, would we still pass?
You don't have to. I strongly recommend that you do at least 12 homeworks and 12 quizzes.
Okay. Thank you.
It just, honestly, Professor Frazier, it makes it difficult because I took this class before, so I kind of already know what's going on. But
okay,
when it comes to like the lab part, I just get so like lost because we don't really learn anything because it's so fast. And like I said, it's only like 15 minutes worth of lecture, or like 20 minutes worth of lecture. And then it's like, well, we're expected to take an exam.
I don't even know how they're conducting that. You know, when I was in school, you know, the lab, you had to see things. And so you're not able to see things. You're not able to see, you know, the equipment you're using. It's like it's all conceptual, if you know what I mean. Like early computer games were text driven, and I had a friend who showed me one of the early games, and I had no idea what he was talking about. You would type in something, and he would say, "Okay, you've just entered this room," and then I'd go, "But you can't see anything. There was no images, you know." And then said, "Okay, you have to decide," and then he would tell you, he says, "Okay, there's a ladder in front of you." And I'd go, "I don't know what," you know, you understand, you can't see anything. So I think that makes it very difficult. It has to be tactile. You have to see things, you have to touch them. That's what's so important about a lab. So I fully understand it could be very frustrating. But I would suggest trying to engage the instructor, maybe, because I'm guilty of doing it too, going a little too fast, and talking to them about slowing down, and also maybe talking to the department head, Dr. Malaki. That's about
We've tried to talk to him about slowing down. He don't care nothing what we got to say.
Well, I'll send an email to Dr. Malaki, tell him that I'll basically tell him that my students are very concerned with the pace of the lab and are having trouble. And could the instructors kind of take that into consideration? Okay,
Yeah,
so
because honestly, from what I was told last semester, I was told that the science department agreed as a whole that the lockdown browser with the camera wasn't necessary because some people couldn't afford it. But he forces us to try to do that, and sometimes my camera doesn't work.
Yeah, I'm sorry, it's just not something I can solve directly for you. But I'll send an email and express some of your concerns to Dr. Malaki, see if that helps. All right, so we have to get back to this. So polymerase chain reaction, it's basically like a Xerox machine for DNA. It rapidly increases the amount of DNA in a sample without the need for culturing or carrying out complex purification techniques. You can replicate a target DNA from a few copies to billions in just a few hours. It's very sensitive. You can detect cancer from a single cell. You can diagnose an infection from a single copy of the gene. So the pooled RT-PCR test they do for COVID, if there was just one piece of the genetic material of the virus, you could detect it using this technique. So what polymerase chain reaction is it takes advantage. This is our DNA replication. It uses the same series of events in DNA replication. Remember, the helicase cuts the bonds. You have a DNA polymerase. You have a continuous strand and a lagging strand with these Okazaki fragments. And so all this that's naturally occurring, these enzymes, we can buy these enzymes in a lab. We can replicate this in a laboratory condition. We can add a mixture of nitrogenous bases that are needed. Everything can be replicated in a lab. So you need some primers. And these are synthetic oligonucleotides, usually 15 to 30 bases. They help indicate where the amplification should begin of the DNA. And you need the DNA polymerase. But you have to do this at high temperatures because you have to separate the DNA. The DNA is separated at a high temperature. So you use special DNA polymerases from thermophilic bacteria. If you remember, these are bacteria that thrive in very hot. Philic means loving. They love hot temperature, and they have an enzyme that isn't affected by the temperature. And so you have a piece of equipment called a thermocycler. And what you're basically doing is you identify a segment of DNA you want to target. You open up the DNA. That's called denaturing. That's your first step. Usually between 50 and 65 degrees Celsius. You add a primer that recognizes nucleotide sequences on the other side of the gene. And then you basically hook these together and you just repeat this process over and over. And you're only synthesizing just a short fragment. That's all you need. You could have a really long gene and it only takes a very short fragment to actually identify that gene. And this process of heating up, separating, reannealing, synthesizing, it's just repeated over and over again. A thermocycler, you might do that 25 or 35 times. It happens over several hours. You have to add a DNA polymerase, your primers, and a mixture of the nucleic acids. It goes through the denaturing step, separating it, priming it, adding the primer, and then you extend it, make your piece right here. You also have another type of polymerase chain reaction. It's called real-time PCR. It can detect products during the reaction instead of at the end. So in other words, you don't have to wait till the very end. You can actually detect them during the reaction. PCR can be adapted to analyze RNA. So this is just DNA here. What you do when you analyze RNA, for example, if you want to detect the COVID, which is an RNA virus, you have to take reverse transcriptase and convert that RNA to DNA with the reverse transcriptase enzyme. And then everything after that is the same. You have a complementary piece of DNA. PCR plays an essential role in mapping genes. That's where the location of a gene is on the genome, or on the chromosome, I should say. You can study genetic defects. It can be used in forensics, infectious disease diagnosis, it's just one of the tests they use, these pooled RT-PCR. On campuses, they can actually take the sewage basically from a dorm and see if they detect any COVID, and then they would know that somebody in the dorm. At my work, it's a pooled sample, so if I've never had a positive test, if it came back positive, then me and everybody who was part of that pool would then have to get individually tested to see if, to find out who's infected. Taxonomy studies, that's the classification of microorganisms, well, I should say all organisms, what groups they belong to. You can study that. So recombinant DNA technology, you remove genetic material from one organism, combine it with a different, and what you have then is genetic clones. Cloning is the removal of a selected gene from an animal, plant, or microorganism, which would be the genetic donor. It's inserted into a vector, which is usually a plasmid or a virus. And then you clone it into a host, usually a bacterium or yeast. The gene is translated into a protein product, which it codes for. DNA is removed from cells and separated into fragments. Whoops, did we skip something here? And separate into fragments, we've already talked about this, cut up by those endonucleases. The gene can be synthesized from isolated messenger RNA by using reverse transcriptase to give us complementary DNA. And then the gene can be amplified by using PCR. Genes maintain in a cloning host in a vector, just like you would in if you had a pure microbial culture. Collection of cDNA clones can represent the entire genome of numerous organisms. So in other words, if we took message RNA from you and made complementary cDNA, we would have a pretty good idea what all your genes are. Plasmids. So if you remember the plasmids, those very small extra chromosomal pieces of DNA in bacteria, they can be transferred in appropriate host cells. We use the same process that the bacteria use called transformation. You can take a cell and shock it or heat it up, and then it will absorb the plasmid. The plasmids can carry genetic markers for resistance, so you can grow your culture in the presence of an antibiotic. And if it's resistant to the antibiotic, you know your plasmid was successfully taken up. You can use a bacteriophage. It will, if you remember, that was our transduction. It has the natural ability to inject DNA into bacterial hosts through transduction. Important aspect when you create a cloning vector. So you can buy these cloning vectors and they can have, these cloning vectors can have established sites for these different endonucleases. These are just examples, and you can buy all these enzymes from a company. You can buy the vector, and then all you would know that, like, for example, if there was a site on here that I could cut with E. coli 1, I can buy this vector, buy the enzyme, make a cut. I can buy it with an ampicillin resistant gene already there. And I can make a cut, put some other DNA into that, put it in a cell culture. And I'm trying to move this around. Okay. Typically contains a gene that confers drug resistance, which is what I said before. So all of this is not something that's naturally occurring. It's made in a lab. Parts of it would have the original plasmid DNA, but you insert a gene. Well, the plasmid could have a gene for resistance, and you would keep that. Microbial cloning is rapid turnover, fast growth. You can grow large quantities using ordinary culture methods. It's non-pathogenic. The genome is well delineated. So in other words, you would know all the genes on the plasmid, capable of accepting plasmids or bacteriophage as vectors for a foreign gene. You can maintain foreign genes through multiple generation, and you can put those into situations where they'll express the proteins. In an industrial setting, you can harvest the proteins, make the bacteria, make them make an antibiotic for you, make a vitamin for you, grow up large quantities and isolate it. Some examples of recombinant DNA technology. For example, the production of alpha-2A interferon. It's used to treat chronic hepatitis, used to treat hairy cell leukemia, and Kaposi sarcoma. The human alpha interferon gene obviously would be put in a vector. It was first isolated and identified in human blood cells. And you use the same processes where you get the messenger RNA, which doesn't have the introns, which you don't need, to eventually make your protein, the alpha-2 interferon. Using recombinant DNA for gene cloning. So you take a genetic donor, isolate a gene, put it in a plasmid. The gene you're inserted, in this case, would be the interferon gene. You isolated it, and then you put it into a cloning vector, put that into a culture, and you can grow it up and harvest your interferon product. The first step in cloning, remember, we use those endonucleases. They cut at specific sites and leave you with sticky ends so that you can insert a gene here. You can then put that into a plasmid, put the plasmid into a bacteria and grow cultures of the bacteria. And the bacteria will make proteins from your cloned vector that you put into it. So once it's inside the cell, they're going to make the message RNA. They're going to translate it for the protein. As I said before, you can just grow up huge batches of these and make the proteins, and then you're going to purify it to where you have your protein product, whatever that protein product of interest is. So DNA can be cut into smaller pieces by using enzymes called what? What are the enzymes called? Called restriction endonucleases. cDNA is produced using the enzyme. It's the enzyme found in HIV virus, called reverse transcriptase enzyme. True or false, PCR uses specialized DNA polymerase adapted to high temperatures. That's true. Remember, you have to have, because you're going to heat up the DNA and separate it, you have to have a DNA polymerase that can stand the high temperature. 2 cloning vectors. We mentioned using plasmids, and we mentioned using bacteriophages in cloning host, usually yeast or bacteria. So products of recombinant DNA technology will provide some examples of recombinant products that have contributed to human health. We're going to list some examples of genetically modified bacteria, plants, animals, and a purpose for each. So anytime you introduce new genetic material into an organism, it becomes a recombinant organism. These can be a source of protein products, nucleotide sequences, medications that cannot be manufactured by other means. We use them in the large-scale manufacture of hormones and enzymes like insulin and human growth hormone. Examples of current protein products from recombinant DNA technology: interferons. So interferons, what they do is they signal cells to fight virus infections, also certain types of cancer, even multiple sclerosis. Interleukins, these are cytokines. They are chemicals that's secreted by the immune system. These cytokines will help fight cancer. It's actually a type of a cytokine, tumor necrosis factor. It can be used to treat cancer also. The brand drugs Remicade and Humira, these are biologics used to treat autoimmune disorders like rheumatoid arthritis and Crohn's disease. You see a lot of these biologics now being advertised quite a bit on TV. And we're able to do that because of DNA technology. Examples of current protein products from recombinant DNA: erythropoietin. It's a peptide that stimulates the bone marrow. It's used to treat forms of anemia. Human growth hormone. It stimulates growth in children with dwarfism. There's also something called wasting syndrome that helps fight that. Some more protein products. Pulmozyme is used to break down the thick lung secretions of people with cystic fibrosis. Tissue plasminogen activating factor can dissolve potentially dangerous blood clots. And PEG SOD is a form of a superoxide dismutase that minimizes damage to the brain and other tissues after surgery or severe trauma. Vaccines that are made. Somebody might need to mute their microphone. Vaccines, hepatitis B, human papillomavirus, Haemophilus influenza, which is a type of meningitis. Some miscellaneous. Factor 8 is replacement blood clotting factor in people who have type A hemophilia. Transgenic organisms, they're anything that's genetically modified. GMOs is transgenic, can be plant or animal. They've had genes introduced into them. Bioremediation is using recombinant microbes. They can be used to clean up oil spills, degrade pesticides and toxic substances. Other uses of recombinant microbes prevent ice crystals on crops, destroy invading insects, production of biofuels, production of new drugs with novel properties, production of industrial chemicals in a more environmentally friendly manner. There's a concern that people have that transgenic plants will share their genes for herbicide, pesticide, and viral resistance with natural plants, leading to super weeds. Scientists counter that non-genetically engineered plants and organisms swap genes all the time, and that inserting insect-resistant genes into crops can reduce insecticide use. Another area is transgenic animals. This involves engineering embryos. Hundreds of strains of transgenic animals have been introduced by research and industry. Unlike bacteria and yeast, animals can express human genes in organs and organ systems very similar to humans. Transgenic animals, they can be used, designed to study human genetic diseases. So you put a diseased human gene into an animal and you can study it. These natural systems are used to test new genetic therapies before they're used on humans. Sheep and goat have also been engineered to become factories. They can manufacture human proteins, excrete them in their milk or semen. This process is known as farming, as in pharmacological. Synthetic biology, the creation of new biological molecule and organisms from scratch. Greg Venter created a self-replicating bacterium in 2010. That's over a decade ago, using just 4 nucleotides of DNA. The first time a living replicating cell had been just created from chemicals. Scientists can create precise chemicals to replace those missing in disease, assemble customized immune components, or construct biological molecules that can precisely target cancer cells. Pathogenic. I know one area of research I've gone to a lecture on it that's very interesting is activating immune cells and getting them to target cancer cells. It's in limited cases, it's actually been very successful in attacking the cancers. The problem so far has been there's a lot of side effects. So far, I don't think any of those therapies have been approved by the FDA. Ethical questions brought up by research into synthetic biology, fears that whatever we create can get away from our control and have unintended consequences. Potential benefits of being able to design cells that create biofuels, medical or agricultural products outweigh hypothetical concerns, though that's a little bit dismissive in my opinion. So 3 products produced through, list 3 products through recombinant DNA technology. We mentioned a whole bunch of those. the alpha interferon. In general, a lot of other cytokines and interferons, tumor necrosis factor, antibiotics. The bacteria Pseudomonas syringae has been modified to prevent the formation. It didn't actually tell you this in this slide, but it's the formation of ice crystals on plants. Transgenic sheep and goat produce human proteins in their milk or semen. And we mentioned before, a scientist over a decade ago basically created an artificial cell. Let's see where we're at. So slide 52. Usually these go to about 100. But I think this goes in a little sooner. So we go up to, we have about 30 more slides. I'm sorry, I should have kept track of where we were. We were around slide 50. All right, so we have about 30 slides left. So genetic treatments, introducing DNA into the body, differentiate between somatic and germline therapy. So all the cells in your body that aren't involved in the creation of sex cells are the somatic cells, and the germline cells are the cells that make the zygote that leads to a baby. Described microRNAs. These are, well, we'll describe what they are in a second. They can their discovery can impact human disease. For certain diseases, the disease phenotype, that's what you actually see, is due to the lack of a single specific protein. So for example, people with type 1 diabetes, they lack insulin. Insulin was first provided from the pancreas of pigs or cows, but we can now make recombinant human insulin. It was the first genetically engineered drug approved for humans. Gene therapy is where we attempt to repair or correct a faulty gene in humans suffering from a fatal or debilitating disease. The benefit is permanent cure of a physical dysfunction by repairing the genetic defect. A normal gene is cloned into retrovirus or adenovirus vectors. Tissues are removed from a patient, incubated with the genetically modified viruses to transfect them. Transfected cells were then introduced into the patient's body. Naked DNA or virus vectors directly introduced into the patient's tissue. So so you're not really getting an infection. The infectious part of the retrovirus, adenovirus, is removed. Some of the vaccines use this too. I'm not sure about the Johnson & Johnson one. That might be a modified adenovirus expressing the COVID proteins. I should look that up. So for an ex vivo type of gene therapy in humans, a normal gene is isolated from a healthy subject. Gene is cloned. The gene is reintroduced into a retrovirus vector. Bone marrow is taken from the patient that has a genetic defect. And the marrow cells are infected with the retrovirus. and the transfected cells are reinfused into the patient. And then you observe the patient, see if they start expressing the normal gene. Experimentation and clinical trials of gene therapy are carried out on human volunteers with a particular genetic condition. Most of these experiments are targeting cancer, single gene defects, and infections. Most gene deliveries are carried out by virus vectors. Early trials hampered by safety issues in which the virus is used to deliver the vehicles actually cause malignancies themselves. Another approach is germline therapy, where you start with the egg cell, the sperm, or an early embryo. The gene is presented in all cells. So then the new gene, if you insert the gene at this very early stage, the new gene is present in all cells of the individual as they go through embryogenesis. And the therapeutic gene is then inherited. So the individual, when they're born, they now have the correct gene and they can pass it on to their offspring. Disease resulting from inappropriate expression of a protein. Some examples: Alzheimer's disease, various viral diseases, many cancers. Sometimes we see this occurs due to malfunctioning what we call microRNAs. So DNA technology as a genetic medicine, cells use a variety of these microRNAs to actually turn off genes, silence their expression. These microRNAs, these are not involved in protein synthesis or translation. They have a completely separate function, and it's regulating gene expression. So we can use microRNAs to fight disease, introducing specific microRNAs into small cell lung cancers, and mice have blocked tumor growth. MicroRNAs have also been used to cause a regression of liver cancer in mice. Using microRNAs to fight disease, inhibiting specific microRNAs in mice have stopped the growth of ovarian and breast cancer. MicroRNAs have been used in African green monkeys to lower their plasma cholesterol levels. Multiplication of hepatitis C in chimpanzees was inhibited by using also a class of microRNAs. But remember, these are all been done in animals, mice, and monkeys. The first successful application in humans. Healthy human volunteers were given a nasal spray containing a microRNA to silence a gene for RSV. Volunteers were infected with RSV and then used the spray for 5 days. 44% of those using the microRNA were infected, compared with 71% of the control. So though it did reduce the infection by this virus, which isn't bad, you're still having a lot of people who still are infected. But on the plus side, those who still were infected, their symptoms were significantly reduced. And that's the case with the current COVID viruses, though that's not microRNA technology, but people are still protected. Even if you get the COVID, you're still protected against serious illness and death by having the vaccine. Name 2 viruses used in gene therapy. We mentioned the adenovirus. What was the other one? Well, I know they use bacteriophages, but those only infect bacteria. I don't remember what the other virus was. True or false, naked DNA is often used in gene therapy. Actually, I'm not quite sure what they mean by that. In gene therapy, you're basically introducing DNA, but you're using a vector to introduce it. So I think that's not true. True or false, there have been no successful human trials for gene therapy. We just saw an example there where there have been some, but it's mostly still something being studied in the lab. Blank therapy describes gene therapy introduced into egg, sperm, or embryo. I actually want to go back and look at that. Germline gene therapy. But I actually don't know if there have been successful human trials of gene therapy. Gene expression can be silenced by these microRNAs.
Is the development of insulin considered one of those?
I'm sorry, which one?
Like the development of insulin from pigs to humans?
Like yeah, actually, that would be that's actually a good example where we use recombinant DNA technology. So the insulin now, instead of the insulin we used to get was from the pancreas of animals, but we no longer use that. We can make recombinant interferon and then just grow it up in bacteria and harvest it and use, and then so then the people are actually getting an interferon that's like human interferon, not like the animal's interferon, which could be potentially a little different.
There's in their HIV stuff as well, and not to mention what they're using with COVID now with the interferon therapy for the cytokine storm.
For the, I'm sorry, for the what?
For the COVID, the interferon infusion for the cytokine storm.
Oh, correct. So interferon can regulate inflammation. So I'm actually not familiar with their treating. So what happens, and this can happen in the flu, something we'll probably mention in the next lectures, people get an overreaction of their immune response. And like you said, it's called a cytokine storm. So they get an overexpression of all these different molecules that increase inflammation. It can be hundreds of them, and they get it's a way overreactive immune response. So I wasn't aware that they're treating that with interferon.
Yeah, it was originally experimental, and what they were seeing was that it was happening in healthy people. So the 23-year-olds who were dying, they were basically dying because their immune system was good.
Yeah,
so
and they were overreacting.
Yeah.
Yeah, so I haven't heard much about that treatment. I know two other treatments. One originally they were doing is people that had been infected. This involves antibody. We're using plasma, and they were trying to harvest their antibodies. And apparently that therapy hasn't panned out. But the other therapy, where a company made monoclonal antibodies to the COVID virus, that's another DNA technology. And the patient gets an infusion, and it gets rid of the virus. That's what Trump got when he got sick with the COVID. And it works really quite well. We had a friend who actually got infected, and she'd previously had problems with becoming septic after getting the flu, and she was a high risk, and her doctor had her, and she got 2 infusions of the monoclonal antibodies, and it worked really quite well. So in this section, we're going to talk about genome analysis, maps, and profiles. Talk about DNA sequencing. We can, with automated sequencers now, we can sequence DNA very rapidly. It takes no time at all to sequence the COVID. It's actually RNA, their genetic materials, RNA. They only have about 15 genes to their genome. We can do DNA profiling, discuss the significance of single nucleotide polymorphisms, referred to as SNPs, in DNA analysis, and describe the utility of DNA microarray technology. DNA can give analysis. DNA can give more accurate mechanisms differentiating between organisms, can identify an organism no longer present that's extinct. Example, when a criminal is identified by DNA extracted from a hair left at a crime scene, can identify a person at increased risk of cancer, Alzheimer's disease before symptoms even arise. Sequence maps, these are detailed maps of the human genome. It can tell you the exact order of bases in a plasmid, chromosome, or the genome. So far, 2,800 viruses, their genomes have been sequenced. When the COVID was first isolated in China, I think it was back January or February, the Chinese actually published the sequence. I know somebody in my lab, he downloaded the sequence, and I remember he was sitting in front of his computer going, oh, this virus has a lot of bat sequences in it. And he was looking at the COVID sequence. There have been 2,000 bacteria and archaeobacteria that their genomes have been published, and 160 different eukaryotes have been sequenced. And of course, the human genome has been sequenced. The head of NIH, Francis Collins, played an important role in that sequencing of the human genome. So the sequence, you can first take a microbial chromosome. If we were starting with a microbe, ultrasound, it fragments the DNA. The fragments are separated on a gel through electrophoresis. We mentioned that before. Each fragment's then inserted into a plasmid and cloned into E. coli. The plasmids are then isolated, purified. DNA fragments are then sequenced by automatic sequencers. So you'd have all this DNA, you grow it up in a bacteria, and then isolate that again and sequence each one. Whole genome sequencing, a computer program can then, so I think originally people had to do this more by sight, but now you can have a computer program written. It will take all the sequence data, is able to find where the sequences overlap. Contigs are large, contiguous, I should say, sets of nucleotide sequences produced by the automated process. The computer can then put them in an order to determine the entire sequence. Editing, a human examines the sequences after they've done all this and looks for irregularities, frame shifts, and ambiguities. So it does require some human analysis at the very end, but this can all be a very speedy process. High throughput sequencing, sophisticated computers and systems develop to sequence genomes in shorter periods of times with less human effort. You can take a sample, isolate the DNA, make the fragments. You add adapters onto the ends of the fragments. You can then use PCR instead of growing them up, as we had on the previous slide. You can PCR them and basically amplify them. You then have millions of amplified sequences, and you have to put them in the same order, look for overlaps, and use software and look at the entire sequence of the genome. Genomics and bioinformatics manage the data produced through high throughput screening. I know at one point they had so much sequence data that researchers have been looking for computer people that can design programs to help them sequence all, because you can now do this so rapidly and come up with a sequence, and you need computer programs to analyze it, to analyze and classify the genes. You can then, with knowing the genetic code, we can then determine protein sequences and ultimately determine the function of the genes. Determining the functional information of a gene sequence, well-annotated genomes provide a complete understanding of what a normal cell's function would be, of disease development, and of aging. These well-annotated genomes will allow us to characterize genetic mechanisms behind pathogens, and allows for the development of new treatments. DNA profiling is a unique picture of the genome. It's also called DNA fingerprinting. This was a forensic science method developed in the 1980s. It's based on the principle that the way nucleotides are combined are different to each organism, or in the case of forensics, to each individual person. It can be used to differentiate between organisms that might be causing a disease outbreak. In criminal investigations, you can match the DNA left at a crime scene. These DNA profiles are like a barcode for life. So samples, if you had samples, different DNA samples, let's say here you have 4 different DNA samples, you fragment them, run them on a gel, and then visualize the bands, and you can see each of these. Let's say this is 4 different individuals. It's going to be a distinct pattern for each one. This is an actual picture of one, and you can see this is the marker. So you have you buy a marker that has known sizes, and you run that along with your samples. And these in the middle here would be the samples. This would be a marker, that would be a marker, that, and you run your samples, and you can look at for differences in the different bands. So for example, they have suspect 1 and 2 and evidence 1 and 2. So if we look at suspect 2, we see 2 bands here, a band here and here. When we look at this individual, we see a band here and here, a band here, and 3 bands here. Down here, we don't see a match to suspect 2, but suspect 1. Evidence 1 and 2, there's a band here, and here. This band that showed up doesn't come up very clearly, but the top 3 bands do. And then you obviously have to run the victims and make sure that's a separate pattern. And the victim here has a separate pattern from all of these. So that's how you would do it in forensic science. We go back to our restriction enzymes. The restriction enzymes, as we said before, cut DNA at specific recognition sites. If a given strand of DNA possesses the recognition site, it will be cut into two pieces. If the same strand from a different person does not have the restriction site, it will not be cut and will remain a large piece of DNA. So each of these two samples is digested with an enzyme and separated on a gel. The first will display two small bands, while the other one might display then one large band. All methods of DNA fingerprinting depends on some variation of this strategy. Now, single nucleotide polymorphisms is important in individual traits. Only a single nucleotide is altered. Between the individuals, tens of thousands of these differences at a single locus exist throughout the genome. Scientists are mapping these single nucleotide polymorphisms to determine which ones put humans at greater risk of disease. Many companies will map these and give a person an estimate of their risk for a variety of disease. You can now send a sample to a company to do that. Examples of single nucleotide polymorphisms associated with disease. There's this protein blood clotting factor 5. Is inhibited by another protein. And so if you have normal clotting, you would have the arginine glycine. Here, what happened is the person, instead of having this arginine here, they have a, I forget what amino acid is this, but it's a different amino acid. And they get excessive clotting because of that single difference. You see the genetic code was GCT, and here it was the C, the cytosine here was replaced by a T, so that would be a single nucleotide polymorphism, and it resulted in a different amino acid. DNA microarray analysis allows biologists to view the expression of genes in any given cell. So you can take the RNA and do reverse transcriptase to get the cDNA and label them with fluorescent dyes. And you put them on these plates, and the fluorescent dyes, when a laser's on them, will show up a pattern. And then you can look at what the colors would represent as different levels of expression of the gene, and then you can analyze it with the computer and see which genes are being upregulated and downregulated. Use of microarrays, developing sensitive diagnostic tests that search for patterns. So this isn't just one gene, you're looking at a whole pattern of gene expression. Being able to identify a subtype of cancer, allowing doctors and pharmacists to treat each cancer with a drug that will be most effective. The steps involved in whole genome shotgun sequencing. The blank genomes provide a complete understanding of cell function, disease development, and other tissues and other issues. The whole genome, I guess. DNA profiling depends on restriction endonuclease enzymes. In situ, I think that technique allows scientists to view gene expression in any given cell. I believe that could be the last. Oh, proteomics. So there's just, so proteomics, so we've been talking all this time about genomics, the expression of genes. And another way to look at it is now they're studying proteomics, the expression of all the proteins. And so with the technology, your what's called your proteome instead of your genome in a cell changes rapidly. And we can see when we compare individuals throughout their life, the genes expression changes, but the proteins they have that are characteristic of certain things changes too. And we can now study that. Cells exert control on translation as well as transcription, and understand gene expression isn't sufficient. So you might have genes, but what you really want to know about is what proteins are actually being expressed, and looking at those. And you have groups of proteins that controls what we call biochemical pathways and different things, and throughout your life that changes. So that's what proteomics is. Proteomics analysis studies the proteins that are produced by a cell at any given moment. A cell can control translation as well as transcription. This says list 2 modern methods of characterizing a proteome. I don't think they actually tell you what those are. So next week we are going to talk about viruses and